We're here at Rothamsted Research to have a look at some really quite exciting cereal crops. So that's both wheat and barley that have got some quite special properties. And they're all part of the Probity project, looking at bringing precision-bred crops to the first commercial farms in Europe. So there's a wheat that has got some very special properties in terms of the health benefits that it then produces for food products. We've also got a barley that produces a high-lipid forage that could have really exciting benefits in terms of methane emissions from cows. So let's go and talk to the scientists and find out. Well, we're here in one of the glass houses at Rothamsted Research, and we're here with some barley plants. And here to explain the barley plants is Professor Peter Eastman. So tell us about this barley. What's special about it, Peter?
Okay, so this barley has been genome edited. It's been altered so that the leaves accumulate more lipid in their cells than wild type, than the normal form of barley. It has a benefit in terms of the added energy content that should help with livestock productivity, lower costs for farmers in terms of their feed costs. The increase in lipid, as I said, should help with reducing methane emissions. And the lipids that are in there are also good for the health of the livestock, and they can affect the quality of the meat and milk that are produced in a positive way as well. So there are multiple benefits.
Fantastic. Let's talk about the actual edit that you've done here, Peter, because it must be the holy grail for forage plant breeders to make a forage that is high in lipid, high energy content, more productivity and so on. Why hasn't that been done before?
So there's certainly a history to this trait, and there's been a lot of interest in it for a number of years. And there are groups around the world that are working on this target. Through conventional breeding and also through using genetic modification. And there's been some success in the context of genetic modification, but of course, those GM crops, it's unlikely that they will ever be grown commercially. In this particular lines, we've targeted the genes that break down oil, and we've basically broken them. And so what happens is the leaves continue to make lipids at the same sort of rate, but their rate of breakdown is slower, so it just gradually accumulates.
Now, one thing that strikes me here, Peter, is that we're looking at barley, and as a farmer I know that barley isn't the first forage crop that a farmer will use. Generally, it's perennial ryegrass. So why have you chosen barley and not perennial ryegrass for this edit?
So yes, you're absolutely right. So I mean, barley is a durable crop, and it can be used as livestock feed. But practically, to have an impact with this particular trait, to make it most useful for farmers, it really needs to be in a forage grass species like lowland perennial, like ryegrass. But barley is much more tractable species to work with. So it's easier to test whether this technology works. In a cereal grass like barley or perhaps like wheat, and then if we can establish that it works reliably, then we can translate this into ryegrass, and this is something that we're very interested in doing.
That's great. Well, let's talk about the actual project itself and what we're hoping to do, because one of the aspects about this project is that we're bulking up these plants, and we've got how many plants have we got in the room here?
I think we've probably only got about maybe 100, 150 plants.
Right, so we've got quite a bit of bulking up to do before we've got enough to actually put out into the field, haven't we?
So that's true. So as a research scientist, obviously we work largely with small numbers of plants in the glass house.
So how soon do you think we'll have enough material to just look at the silage, look at the actual content of the silage? For
next season, obviously, it's a spring variety, this variety of barley. So we will plant it in the spring, and I guess by the summer, autumn, we will have material grown on a scale that's sufficient that we can start doing that analysis.
One of the aspects of the research that you've looked at, and in terms of the genes that you're working with, is that you've been able to get a gain of function within the plant. Could you just explain what a gain of function is and how you've managed to do that?
Yeah, sure, I can. So we've used the genome editing technologies basically to break genes, to stop genes working, and this is quite technically easy, and you can achieve a whole range of useful outcomes from doing that. But a lot of interesting traits that you can introduce into plants, you know, are caused by gain of function, by genes increasing their activity or changing the patterns through development or spatially within the plant or other organism, you know, when they're active. It's harder to achieve those sort of changes using genome editing technologies, but there are tricks and ways of doing it.
Peter, one of the aspects about the Probity project is that we're bringing gene-edited crops, and specifically your barley, onto farms, commercial farms, for the first time. What's the significance of that for you?
I think it has huge significance. I mean, obviously it's the first time for me that such a thing has become possible or an option within my research career as an academic researcher. But I mean, everything that we do, and in the context of this material, it's key basically that the material is tested on farm, in the field. And it's key to understand whether farmers can grow it, otherwise it will have no utility. It's so important that we move forward and we test things in the field, and we test things with, well, all the way through potentially the chain to understand whether these things will stand up or not from a commercial point of view. Otherwise, you know, we just won't know what the potential is, and it will never be realised. So for me, it's very exciting to participate in this project.